In this video, we are going to talk about the cell wall of the bacteria. The cell wall is the outermost component common to all bacteria except Mycoplasma, which are bound by a cell membrane only. Some bacteria have surface features external to the cell wall such as capsule, flagella and pili, which are less common components. The cell wall is located external to the cytoplasmic membrane or the cell membrane, and it is composed of peptidoglycan. The peptidoglycan provides support and maintains the characteristic shape of the cell. Let's talk about the cell walls of gram-positive and gram-negative bacteria. The structure, chemical composition and thickness of the cell wall differs in gram-positive and gram-negative bacteria. The peptidoglycan layer is much thicker in gram-positive than in gram-negative bacteria. Many gram-positive bacteria also have fibers of teichoic acid that protrude outside the peptidoglycan, whereas gram-negative bacteria they do not have any teichoic acids. In contrast, the gram-negative bacteria have a complex outer layer consisting of lipopolysaccharides, lipoproteins, and phospholipid. Lying between the outer membrane layer and the cytoplasmic membrane in gram-negative bacteria is the periplasmic space, which contains many different hydrolytic enzymes such as beta-lactamases that degrade penicillins and other beta-lactam drugs. The cell wall has several other important properties as well. In gram-negative bacteria, it contains endotoxin, which is a lipopolysaccharide. The polysaccharide and proteins of the cell wall, they serve as antigens that are useful in laboratory diagnosis. The cell walls also contain some specific proteins called porin proteins, which facilitate the passage of small hydrophilic molecules into the cell. Porin protein in the outer membrane of gram-negative bacteria act as a channel to allow the entry of essential substances such as sugars, amino acids, vitamins and metals, as well as many antimicrobial drugs such as penicillins. Cell walls of acid-fast bacteria, Mycobacterium, have an unusual cell wall resulting in their inability to be gram-stained. These bacteria are said to be acid-fast because they resist decolorization with acid alcohol after being stained with carbolfuchsin. This property is related to the high concentration of lipids called mycolic acids in the cell wall of Mycobacteria. In view of the importance, there are three components of the cell wall: peptidoglycan, lipopolysaccharide and teichoic acid. Now we will discuss each of these one by one. Peptidoglycan. Peptidoglycan is a complex interwoven network that surrounds the entire cell, and it is composed of a single covalently linked macromolecule. It is found only in bacterial cell walls. It provides rigid support for the cell. It is important in maintaining the characteristic shape of the cell, and it allows the cell to withstand media of low osmotic pressure. A representative segment of peptidoglycan is shown in this diagram. The term peptidoglycan is derived from the peptides and the sugar, glycan, that make up the molecule. Synonyms of peptidoglycan are murin and mucopeptide. This figure illustrates the carbohydrate backbone, which is composed of alternating N-acetyl muramic acid and N-acetyl glucose amine molecules. Attached to each of the muramic acid molecule is a tetrapeptide consisting of both D and L amino acids. The precise compositions of these amino acids differs from one bacterium to another. Two of these amino acids are worthy of special mention: diaminopimelic acid, which is unique to bacterial cell walls, and D-alanine, which is involved in the cross links between the tetrapeptides and in the action of penicillin. The other important component in this network is the peptide cross-link between the two tetrapeptides. The cross-links vary among species. In Staphylococcus aureus, for example, 5 glycines link the terminal D-alanine to the penultimate L-lysine. Because peptidoglycan is present in bacteria but not in the human cells, it is a good target for antibacterial drugs. Several of these drugs, such as penicillins, cephalosporins, and vancomycins, inhibit the synthesis of peptidoglycan by inhibiting the transpeptidase, which is the enzyme that makes the cross links between the two adjacent tetrapeptides. Lysozyme, an enzyme that is present in human tears, mucus, and saliva, can cleave the peptidoglycan backbone by breaking its glycosyl bonds, thereby contributing to the natural resistance of the host to microbial infection. Lysozyme treated bacteria may swell and rupture as a result of the entry of water into the cells, which have a high internal osmotic pressure. If the lysozyme treated cells are in a solution with the same osmotic pressure as that of the bacterial interior, they will survive as spherical forms, which are called protoplasts, surrounded only by a cytoplasmic membrane. Now we will talk about lipopolysaccharides. The lipopolysaccharide of the outer membrane of cell wall of gram-negative bacteria is called endotoxin. It is responsible for many of the features of disease such as fever and shock, especially hypotension, which is caused by these organisms. It is called endotoxin because it is an integral part of the cell wall in contrast to the exotoxin, which are actively secreted from the bacteria. The constellation of symptoms caused by the endotoxin of 1 gram-negative bacterium is similar to another, but the severity of the symptoms can differ greatly. In contrast, the symptoms caused by exotoxins of different bacteria are usually quite different. The lipopolysaccharide is composed of 3 distinct units: a phospholipid called lipid A, which is responsible for toxic effects; a core polysaccharide of 5 sugars linked through ketodeoxyoctolonate KDO to lipid A; an outer polysaccharide consisting of up to 25 repeating units of 3 to 5 sugars. This outer polymer is the important somatic or O antigen of several gram-negative bacteria that is used to identify certain organisms in the clinical laboratory. Some bacteria, notably members of the genus Neisseria, have an outer lipo-oligosaccharide LOS containing very few repeating units of sugars. Lastly, we have teichoic acid. Teichoic acids are fibers located in the outer layer of the gram-positive cell wall and extend from it. They are composed of polymers of either glycerol phosphate or ribitol phosphate. Some polymers of glycerol teichoic acids penetrate the peptidoglycan layer and are covalently linked to the lipid of the cytoplasmic membrane, in which case they are called lipoteichoic acid. Others anchor to the muramic acid of the peptidoglycan. The medical importance of teichoic acid lies in their ability to induce inflammation and septic shock, which, when caused by certain gram-positive bacteria, that is, they activate the same pathways as does endotoxins in gram-negative bacteria. Teichoic acids also mediate the attachment of the staphylococci to the mucosal cells. Gram-negative bacteria do not have teichoic acids.